Hello guys, welcome to another video. In this video we are going to learn about the gene transformation. So what is gene transformation? Gene transfer is the uptake of foreign DNA or transgene by plant cells. It is the subsequent stable integration and expression of foreign DNA into the genome. The method used for gene transfer is known as transformation. Then, what are the methods of transformation? There are mainly two types of methods, such as indirect or direct. Indirect or Agrobacterium is mediated gene transfer. Gene transfer is done by using Agrobacterium bacteria. Then direct gene transfer. By using various techniques listed on next page, the gene is directly transferred into the host. Here we can see the types of genetic transformation. Mainly two types. First one is indirect, which is caused by Agrobacterium-mediated gene transfer, and direct method of gene transfer. Direct method is two types: physical and chemical. Physical, such as electroporation, ballistic, macro-injection, micro-injection, liposome-mediated, etc. Then chemical. PG-mediated dimesopolycation, dextran, calcium phosphate, co-precipitation. So these are the chemical. In this video we are going to learn all of these types in briefly. So let's get started. First one is indirect or Agrobacterium-mediated gene transfer. Agrobacterium is soil-borne, gram-negative, rod-shaped, motile, found in rhizosphere. Causative agents of crown gall in dicotyledons. Two strains of T. plasmid. Octopine strains contains Tdna region T14 kb and Tr7 kb. Then Nopaline strains contains Tdna region 20 kb. The size is about 200 kb, contain a V region 40 kb, at least 8 to 11 V genes. Then here you can see the T. plasmid, which contains origin, then Tdna region, which is auxin, cytokinin, opine. Then this is the V region, VA, which wound signal receptor, VB, membrane proteins, VG, V activator, VC, binding protein, VD, endonuclease, then VE, DNA binding protein, VF, nuclear targeting of ssDNA, single stranded binding DNA, that is VH, host range specificity. Then these are octopine catabolism, replication origin, and incompatibility. So this is T. plasmid, figure of T. plasmid. Then Tdna. Here we can see Tdna. This is Tdna region, which is auxin, cytokinin, opine synthesis. Then Tdna size is 12 to 24 kb, left and right border sequence 24 base pair, which will transfer into a genome of host plant. Here you can see left border and right border. Then oncogenes, that means auxin, cytokinin, opines. The DNA contains 8 potential genes. These are eukaryotic in nature: eukaryotic promoters, monocystonic, eukaryotic polyadenylation signals, eukaryotic translation mechanisms, etc. Then process of Tdna transfer and integration. Identify a suitable explants. Suitable plant tissue is removed and sterilized. Co-cultivate with the Agrobacterium. Small pieces of leaf tissue placed into a culture of Agrobacterium for 30 minutes. The explants then placed on MS medium without selective agent. Incubate explants with Agrobacterium for 2 days to allow transfer of the Tdna. Kill the Agrobacterium with a suitable antibiotic. The explants are removed from the medium and washed in cephalotaxime. Select for transformed plant cells. The explants are transferred to a selective medium with cephalotaxim. Auxin cytokinin are used to encourage the regeneration of by organogenesis. Regeneration of whole plant. The shoot can be rooted by placing them on a solid medium containing high auxin and cytokinin ratio. Then electroporation. Electroporation is direct method, which is physical direct method. Electroporation plant materials is incubated in a buffer solution containing DNA and subjected to a high voltage electric pulse. The DNA then migrates through high voltage induced pores in the plasma membrane and integrates into the genome. It can be used to transform all the major cereals, particularly rice, wheat, maize. It can be used to deliver DNA into plant cells and protoplasts. There are two systems of electroporation: low voltage, high voltage. Low voltage, long pulses, 300 to 400 volts per centimeter for 10 to 50 minutes, produce high rates of transient transformation. High voltage short pulses, 1000 to 1500 volt per centimeter for on 10 microsecond, produce high rates of stable transformation. Transformation frequency can be improved. A pure heat shock treatment to protoplast, presence of low concentration of PEG 8%. The advantages of electroporation: both intact cells and tissue can be transformed. The efficiency of transformation depends upon the plant materials. Disadvantages: 40 to 50 incubated cells receive DNA, 50% of transformed cells can survive. So these are the disadvantages. Then micro injection. A holding pipette holds the protoplast while an injection pipette injects the macromolecule. For manipulation of protoplasts without damage, protoplasts are cultured about 1 to 5 days before the injection. The injection through the partially regenerated cell wall facilitates to target particular compartments to the cell, particularly useful for transformation of plant protoplast with exogenous genes. Here you can see the macro injection. Then this is micro injection and macro injection. Macro injection is the mediate method tried for artificial DNA transfer to cereals plants that show inability to regenerate and develop into whole plants from cultured cells. Needles used for injecting DNA are with the diameter greater than cell diameter. DNA injected with conventional syringe into region of plant which will develop into floral tillers. Here are the advantages of macro injection. This technique does not require protoplast. Instrument is simple and cheap. Methods may prove useful for gene transfer into cereals which do not regenerate from cultured cell easily. Technically simple. And what are the limitations? It is less specific, less efficient, frequency of transformation is very low. So this is all about the macro injection. Then ballistic method, firstly used by Klein et al. 1987 and Sanford et al. 1987, also called as ballistic method, gene gun method, particle bombardment, particle gun method, micro projectile, etc. Gene gun is developed to enable penetration of the genetic material containing a gene of interest in the cell wall. 1 to 2 micrometer tungsten or gold particles are used coated with the DNA. Acceleration is given to enter the micro projectiles into the plant cells. Acceleration achieved by using a device varies in design and function. Devices: pressurized helium gas or electrostatic energy released by a droplet of water exposed to high voltage. Here you can see gas acceleration, macro carrier, discs, stopping screen, microcarrier launch assembly, DNA coated targeted cells and target cell. So this is about the ballistic method or gene gun method. Then what are the advantages of a gene gun method? Advantages: this method can be used to transform all plant species. No binary vector is required. A transformation protocol is relatively simple. Then what are the disadvantages of gene gun or ballistic method? Difficulty in obtaining single copy transgenic events, high cost of equipment and microcarriers. Intracellular target is random: cytoplasm, nucleus, vacuole, plastid, etc. Transferred DNA is not protected. Then liposome mediated gene transfer. Liposomes, which is physical direct method. Liposomes are spheres of lipids used to transport molecules into the cells. These are artificial vesicles that can act as delivery agents for exogenous materials, including transgenes. They are considered as sphere of liquid bilayers surrounding the molecule to be transported and promote transport after fusing with the cell membrane. Here you can see this is DNA and this is liposome. DNA is inserted into liposome and liposome is attracted with the cell and it is inserted into the nucleus. After insertion, this is cytoplasm, and after inserted into the cell, it is known as endosome. And endosome, there are some defense mechanism in cell which cause death of endosome, and some intake some genes are killed by defense mechanism of cell, and some can survive. This survival gene inserted into the nucleus, and DNA is uncoating and DNA transcription occur, and mRNA transfer into the cytoplasm and translation occur into protein synthesis. So this is the indirect liposome mediated method. And direct means directly gene is directly inserted into nucleus, then DNA uncoating and transcription is occur, then mRNA transfer into cytoplasm, and then translation occur and protein synthesis occur. So this is direct and this is indirect liposome mediated method. Cationic lipids are those having positive charge are used for transfer to nucleic acid. Liposomes are able to interact with negatively charged cell membrane more rapidly than uncharged liposomes. Due to fusion between cationic liposome and cell surface results in the delivery of DNA directly across the plasma membrane. Cationic liposomes can be produced from a number of cationic lipids, DOTAP and DOTMA. These are commercially available lipids that are sold as in vitro transfecting agent as lipofectin. So what are the advantages of liposome mediated gene transfer? High degree reproducibility, long term stability, low toxicity, protection of nucleic acid from degradation. Then PEG mediated gene transfer, which is direct chemical method. PEG means chemical method. PEG, polyethylene glycol, in the presence of divalent cations using calcium, destabilizes the plasma membrane of protoplast and renders it permeable to naked DNA. In this way, the DNA enters nucleus of the protoplast and gets integrated with the genome. Culture of protoplast is taken into a tube, and to this tube 40% PEG 4000 W/V dissolved in mannitol and calcium nitrate is added slowly, then incubated for few minutes. So what are the advantages of PEG? Advantages are a large number of protoplast can be simultaneously transformed. can successfully use for a wide range of plant species. And what are the limitations of PEG? The DNA is susceptible for degradation and rearrangement. Random integration of foreign DNA into genome may result in undesirable traits. Regeneration of plants from transformed protoplast is a difficult task. Then calcium phosphate co-precipitation. The DNA is allowed to mix with calcium chloride solution and isotonic phosphate buffer to form DNA calcium phosphate precipitate. When the actively dividing cells in culture are exposed to this precipitate for several hours, the cells get transformed. The success of this method is dependent on the high concentration of DNA and the protection of the complex precipitate. Addition of dimethyl sulfoxide DMSO increases the efficiency of transformation. DNA imbibition by cells. Some workers have seriously tried to transform cells by incubating cell suspension, tissues, embryos, and even seeds with DNA. The belief is that the DNA gets imbibed and the cells get transformed. DNA imbibition approach has met with little or no success. Imbibition, the absorption of one substance by another, in particular the uptake of water by a plant or seed. Then DMSO polycation. DMSO dimethyl sulfoxide can interact with the plasma membrane, allowing pores formation, which contribute to decrease membrane selectivity and increases cell permeability. It involves use of polycation polybrane to increase the absorption. The absorption of DNA to the surface followed by 25 to 30% DMSO to increase the membrane permeability and enhance the uptake of DNA complexes. The major advantages of polybrane is that it is less toxic than other polycations and high transformation efficiency requires every small quantities of plasmid DNA to be used. Then dextran. The decidable DNA can be complexed with a high molecular weight polymer diethyl aminoethyl DAE diethyl aminoethyl. Dextran are transferred. The efficiency increased to 80% when DMSO shock is given. The major limitation of this approach is that it does not yield stable transformants. So these are all about the gene transformation. That's all for today. See you in the next video. Thanks for watching.